The MHC class 1 and class 2 molecules deliver peptides to the cell surface from two distinct intracellular compartments. Infectious agents can replicate in either of two distinct intracellular compartments. Viruses and certain bacteria replicate in the cytosol or in the contiguous nuclear compartment, whereas many pathogenic bacteria and some eukaryotic parasites replicate in the endosomes and lysosomes that form part of the vesicular system. The immune system has different strategies for eliminating pathogens from these two sites. Cells infected with viruses or with bacteria that live in the cytosol are eliminated by cytotoxic T cells. These T cells are distinguished by the co-receptor molecule CD8. The function of CD8 T cells is to kill infected cells. This is an important means of eliminating sources of new viral particles and obligate cytosolic bacteria, thus freeing the host of infection. Pathogens and their products in the vesicular compartments of cells are detected by a different class of T cell, distinguished by the co-receptor molecule CD4. CD4 T cells are specialized to activate other cells and fall into two functional classes. Th1 cells, sometimes known as inflammatory T cells, whose main function is to activate macrophages to kill the intravesicular pathogens they harbor, and Th2 cells, or helper T cells, which activate B cells to make antibody. Microbial antigens may enter the vesicular compartment in either of two ways. Some bacteria, including the mycobacteria that cause tuberculosis and leprosy, invade macrophages and flourish in intracellular vesicles. Other bacteria proliferate outside cells where they cause pathology by secreting toxins and other proteins. These bacteria and their toxic products can be internalized by phagocytosis, endocytosis, or macropinocytosis into the intracellular vesicles of cells that then present antigen to T cells. These include the dendritic cells that specialize in initiating T cell responses, macrophages that specialize in taking up particulate material, and B cells that efficiently internalize specific antigen by receptor-mediated endocytosis of the antigen bound to their surface immunoglobulin. To produce an appropriate response to infectious microorganisms, T cells need to be able to detect the presence of intracellular pathogens and to distinguish between foreign material coming from the cytosolic and vesicular compartments. This is achieved through the use of the different classes of MHC molecule. MHC class 1 molecules deliver peptides originating in the cytosol to the cell surface, where they are recognized by CD8 T cells. MHC class 2 molecules deliver peptides originating in the vesicular system to the cell surface, where they are recognized by CD4 T cells. CD8 and CD4 bind MHC class 1 and MHC class 2 molecules respectively, and so help to ensure that the appropriate type of T cell is activated in response to a given pathogen. And that's the MHC class 1 and MHC class 2 molecules deliver peptides to the cell surface from two distinct intracellular compartments. Have a great day.